continue our journey with reproductive biology of angiosperms once again, and today we will be talking primarily about embryogenesis. We've been talking about fertilization in angiosperms in last few classes, and you know that there is double fertilization, which is a characteristic feature of angiosperms, wherein one male gamete fuses with the female gamete, which we call as syngamy, and the second male gamete, it fuses with the two polar nuclei which are present in the central cell to form the primary endosperm nucleus along with the male gamete. So, end product of fertilization between the sperm and the egg is a zygote, and it is the zygote with which the life cycle of angiosperms begins. This zygote will undergo some organized divisions and ultimately will give rise to an embryo, and this process of development of an embryo from zygote is known as embryogenesis. There is a particular pattern, form and polarity which exists during the development of embryo, and this results from several cellular, molecular and genetic mechanisms. You know that both dicot and monocot embryos, they share the same ontogeny up to a particular stage. A typical dicot embryo comprises of an embryonal axis with two cotyledons attached to it laterally. Now you see here that this is a general structure of an embryo. The part of the embryonal axis above the level of cotyledons is known as the epicotyle. This epicotyle terminates into an embryonic shoot, which we also refer to as the plumule. The part of embryonal axis which is below the level of cotyledons is known as the hypocotyle, and this hypocotyle gives rise to the embryonic root, and embryonic root is something which we call as the radicle as well. In this particular drawing, you can see that a seed has been split open to show the embryo, and you can see that how the epicotyle and hypocotyle are organized, and when a seed germinates, it's always the root which comes out first. You would have even seen, when you put Bengal gram or moong dal for sprouting. So, the white structure which comes out basically is a root. A typical monocot embryo differs from a dicot embryo in having only one cotyledon attached to the embryonal axis, in contrast to two cotyledons which are present in the dicot embryo. In this particular diagram, you can see three different types of embryos. The first one is a typical dicotyledonous embryo belonging to Capsula bursa pastoris. The second one is a monocotyledonous embryo of Hydrilla, and third one is a grass embryo. Now, the cereals and the grasses are also monocots, but their embryos are different from other monocots because they have an additional embryonic organ which is associated with them, and these can be scutellum, coleoptile, coleorhiza. Scutellum is what? Scutellum is a seed leaf. It is a massive cotyledon which covers the embryo, and lateral to this scutellum, that is on one side, there is a short embryonal axis which is divided again into epicotyle and hypocotyle. Epicotyle is above the level of scutellum and hypocotyle is below the level of scutellum. The epicotyle comprises of several young leaves, and these are covered by the coleoptile. The leaves of the shoot apex are covered by a sheath called coleoptile. Hypocotyle consists of root apical meristem, and this is covered by coleorhiza. Coleorhiza can be seen developing into a small outgrowth which is on one side, and this is called as epiblast. You can see a small outgrowth on the right hand side of the shoot apex, and this is the epiblast. Now, zygote of course is an undifferentiated cell at its inception because it is formed by the fusion of male and female gametes. Very soon this zygote undergoes organized changes which occur in the cytoplasm as well as in the organelles. Zygote also undergoes dormancy period which can vary. It can be a few hours or it can be up to several days. In Hippophae rhamnoides, the dormancy period is 50 days, whereas in Capsula it is only about 24 hours. So, shorter dormancy is seen in species which exhibit a cellular type of endosperm as compared to species with a nuclear endosperm. Once fertilization is accomplished, the large micropylar vacuole of the fertilized egg, this is replaced by numerous small vesicle-like vacuoles. So, instead of one large vacuole which is present in the egg cell, in the zygote there are numerous small vesicles which are present. Gradually, all these small vesicle-like vacuoles they fuse and again they form a large vacuole which then occupies about 2/3 of the total volume of zygote, due to which the zygote elongates because the major part of the zygote at this stage is occupied by the vacuole. Formation of vacuoles in zygote, it leads to a major change in the volume of the zygote along with the polarity being created. Now this polarity is created only because of the presence of vacuole. One of the first patterning events after fertilization is the formation of an apical basal axis of the zygote and hence the attainment of polarity. In this one, which is a transmission electron micrograph of an elongated zygote in Nicotiana, you can see that there is a pronounced apical and basal polarity. Now, as the zygote elongates, its polarity is reacquired with nucleus towards the chalazal end and a large vacuole towards the micropylar end. A considerable shift occurs in the volume 2 of the zygote during the period of dormancy. The establishment of polarity in ultrastructural profile of zygote, this results in the formation of two poles, the basal and the apical. The basal pole is the one which lies at the micropylar end of the embryo sac and mostly possesses a large vacuole. The apical pole on the other hand is present towards the callosal end of embryo sac and it has a large number of cell organelles which also includes the nucleus. This polarity of the formation of a basal and an apical pole lays the foundation for future divisions and differentiation of the embryo. We can also say that this is the first step in embryo patterning. So, what you have understood? We talked about the zygote and then we talked about the formation of an embryo. So, the entire process of formation of a mature multicellular embryo from a single celled zygote is what we call as embryogenesis. Attainment of polarity in zygote makes it all set for its first division. The first division in the zygote is invariably asymmetric and transverse, leading to the formation of two unequal sized cells. There is one cell which is smaller and this is the apical cell. The apical or the smaller terminal cell gives rise to the organogenetic parts of the embryo. That means it will give rise to the epicotyle, the shoot apical meristem and all. The larger basal cell gives rise to a suspensor or a hostorium. Now, basal cell, like I said earlier also, is present towards the micropylar end of the embryo sac. It is larger in size and possesses a large vacuole. Cytoplasm is very sparse because the major portion of the cell is occupied by the vacuole and this basal cell is devoid of organelles. The main function of the basal cell is to form a suspensor and also a root apex. The subsequent divisions in the basal cell are transverse. The apical cell is present towards the callosal end of the embryo sac. It is much smaller in size than the basal cell. Vacuoles are mostly absent because the major part of the cell here is occupied by the organelles. Apical cell is rich in organelles, the cytoplasm is dense, and it is the apical cell which forms organogenetic parts like the cotyledons and the shoot apical meristem. Subsequent divisions in the apical cell are transverse or even longitudinal. Remember, I told you that in basal cell the subsequent divisions are transverse. So, here there is an option for an apical cell. It can either undergo a transverse or a longitudinal division. So, depending on all this, that is based on the contribution of apical and basal cells and also on the plane of division in apical cells, the type of embryo development varies. The plane of division in the basal cell is mostly transverse, but the plane of division in the apical cell varies because it can be either transverse or longitudinal. Based on the plane of division, there are different types of embryogenies which exist. So, when we talk about the plane of division, there are three different groups of embryogeny which are observed in angiosperms. The group 1 or the first one is where plane of division in the apical cell is longitudinal. I would like to remind you here that the plane of division in the basal cell is always transverse. So, here you see that the plane of division in apical cell is longitudinal, and we have two types of embryogenies here, onegret and estirad type. The second group is where the plane of division in the apical cell is transverse. So, there are further types of embryogenies which are seen when the plane of division in the apical cell is transverse. The third group is when the plane of division is longitudinal in the zygote. So far in the last two ones we have seen that the plane of division, first plane of division is transverse, resulting in the formation of an apical cell and a basal cell. So, in group 1 and in group 2, the first division is transverse, which results in the formation of an apical and a basal cell. And then depending on the plane of division in the apical cell, the embryogeny differs. In group 1, it is longitudinal, and in group 2 it is transverse. Now, in group 3 there is no formation of apical and a basal cell. The zygote here itself, it divides longitudinally. So, this is the third group of embryo formation, and it is observed in the members of family Viperaceae. Now, plane of division in apical cell when longitudinal, it leads to two types of embryogenies, the onegret and the estirad type. First, I will talk to you about the estirad type of development. You see in this particular diagram, you can see two colors. The light grey, it represents the basal cell, and the black or the dark grey represents the apical cell. So, in estirad type of embryo formation, the embryo is generated from the derivatives of both basal cell and apical cell, which you can see here, with apical cell forming the cotyledons and the plumule, and the basal cell forming roots, root cap, hypocotyle and suspensor. Estirad type of embryo formation is seen in Asteraceae and Poaceae family. Now, you can see here that both the cells, that is the basal cell and the apical cell, they contribute to embryo formation. The second type is the onegret type. Here the basal cell may contribute to the formation of embryo. Okay. So, onegret type is also called as the crucifer type of embryo formation. Here the mature embryo is generally derived from the apical cell alone. The basal cell contributes only towards the formation of a suspensor. It is called onegret type because it is seen in family Onegraceae. Now, one thing again I would like to repeat here, in Esteret type the basal cell is forming roots, root cap, hypocotyle and suspensor. In onegret type the basal cell merely contributes towards the formation of suspensor. Then the second type is when the plane of division in the apical cell is transverse. Now, in all three diagrams here you see what C, D and E, you see that the first division of course is transverse when apical and basal cells are formed, and then later on the basal cell is always dividing transversely, or it may not even divide as you can see in the caryophyllid type, but the apical cell can divide. Now here it will divide, the plane of division is transverse. The first one is the solenid type where the apical cell and basal cell, they both contribute to the formation of embryo. So, you can see that there are divisions occurring in both apical as well as basal cells. In the Chenopodiad type, only the apical cell contributes to the formation of embryo. Basal cell does contribute to the formation of a suspensor. The third or the caryophyllid type, only apical cell contributes to embryo, basal cell does not divide further. So, when both basal cell and apical cell they contribute to the formation of embryo, we have Chenopodiad type. The embryo arises from both apical and basal cells. Hypocotyle of mature embryo is derived from the basal cell, while the epicotyle arises from the derivatives of the apical cell, seen in family Chenopodiaceae. When basal cell does not contribute to the formation, but it does contribute to the formation of a suspensor, we have the solenid type of embryogeny. Embryo is primarily formed by the apical cell. Divisions in the basal cell, when they occur, they contribute to the formation of a suspensor. This type of embryogeny is characteristic of family Solanaceae. Then comes the caryophyllid type, embryo formed exclusively by the apical cell. Basal cell here does not divide any further. If a suspensor is present, it is derived from the basal cell and is unicellular. Why? Because I told you that this basal cell remains unicellular because it does not divide any further. So, this is seen in family Caryophyllaceae. So, I hope you have understood the different types of embryo formation we have here. So, I would like to repeat here that one is plane of division in the apical cell is longitudinal, second plane of division in apical cell is transverse, and third plane of division in is longitudinal in the zygote. In the third group there is no formation of apical cell or basal cell. Now further divisions they take place, so it will matter whether the division in the apical cell is longitudinal or it is transverse. If it is longitudinal, we have the Esterad and the Onegret type, and if it is transverse, then we have three types, that is Solenad, Chenopodiad and Caryophyllid type, and this depends on how much each one of these is contributing. If it is only the apical cell which is contributing, or it is apical cell and some bit from the basal cell, or it is both which contribute. So, these leads to different types of embryogeny. The third one is when the plane of division in the zygote is longitudinal. This is seen in families Piperaceae and Laurentheaceae. First division in the zygote is vertical. So, if there is a vertical division, it does not lead to the formation of any apical or basal cell. So, in piperid type of embryo formation, there is no apical and basal cell which is formed. Instead, two elongated cells are formed, and these two elongated cells, they lie parallel to each other, and they cut off to form the embryo. Okay. Now, there will be further divisions which take place in these two cells, which are formed by a longitudinal division, and subsequent divisions lead to the formation of a full-fledged embryo. Now, what is embryo patterning? We have been talking about the formation of embryos, and of course, I told you that the process of formation of embryo from a zygote is what is called as the embryogenesis. Now, when the embryo formation is taking place, all the cells which are involved in the formation of embryo, they acquire a specific structure and function, and this is what is called as embryo patterning. So, whenever divisions are taking place in the zygote, each cell takes up a different function, it has a different size, it has a shape, and of course, then they have a specific structure. So, when we assign a specific structure and a specific function to a particular cell which is giving rise to an embryo or which is contributing to the formation of embryo, the whole process is called as the embryo patterning. So, basically how the cells they differentiate to perform different functions as an embryo, that is what we call as an embryo patterning. So, you see in this particular picture that the first division is, of course, this is a transverse division. So, there is formation of an apical cell, there is basal cell, and then there are further divisions which occur in each of the apical and the basal cells. You can see that the apical cell is giving rise to two cells by a transverse division, and even a transverse division in the basal cell also leads to formation of two cells. These four cells eventually they divide again and again. So, first there will be formation of so from one cell zygote there is formation of two cell structure, then four cell, and when it undergoes another mitotic division in one particular plane there are 8 cells, which is an octant configuration, and when these 8 cells also divide further, we have a 16 celled pro-embryo which is formed, so on and so on, so forth. So, talking about embryo patterning, you see here that one division each in apical cell and basal cell of the two celled pro-embryo, it results in a tetrad stage of pro-embryo. First division in basal cell is transverse, and the two cells C, I and M occur in single tier at the 4-celled pro-embryo stage, because a transverse division in apical cell also leads to formation of two cells, that is L and L dash. So, this is what embryo patterning is, and I would like to continue with the topic of embryo patterning in the next lecture. Thank you very much for your patience listening. Thank you.